This is TWIS. This Week in Science, episode number 614, recorded on Wednesday, April 12th, 2017. Thank you, Yuri. Hey everyone, I am Dr.Kiki, and tonight on This Week in Science, we are going to fill your heads with evolving octopuses, young fish poo, and magnetic sperm. But first...
Disclaimer, disclaimer, disclaimer. Humans. They seem to be everywhere these days. And while we watch their hopes and dreams, fears and flaws play out across the globe, the wider world is waiting. The world of everything that is not human. That world that has been watching the activities of hairless apes, sometimes with curiosity, too often in horror. The world is watching, waiting for humans to strike a balance, to find a niche within the greater Earthling community. And while the wayward activities of the hairless apes shows little sign of slowing, there are some promising signals, as some of the brighter ones begin to understand that within the mysteries of how things work, there is a lesson in longevity, a logic to living within the balancing act of other life forms. And so, to get the word out to as many as possible, we will take human form just long enough to bring you yet another episode of This Week in Science. Coming up next.
I've got the kind of mind I can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
And a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We're back yet again with more science, another week filled with the fantasticness of the science world. You know what tonight is?
It's Wednesday, right?
It's Wednesday.
Okay.
It's also Yuri's night.
Ooh.
Tonight is Yuri's night. And if anyone is aware of what Yuri's night is, maybe you're out celebrating right now as we record this show, or, maybe you're celebrating it in your own way in your own home by watching
live in
this
suspense
show.
any longer. Kiki, what am I not celebrating that I should be out celebrating with people who know about this event?
The instance of the first human in space, Yuri Gagarin. This is the anniversary of Yuri Gagarin's space flight. He was a Russian cosmonaut, the first man in space. And there are celebrations all over the world in honor of Mr. Gagarin, cosmonaut Gagarin. Yes, not Yuri Geller, Gagarin. So this is an auspicious day in the history of spaceflight, manned human spaceflight, which, there's also womaned human spaceflight. And as you may well know, there is an astronaut at the International Space Station who she is on her way to extending her stay on the International Space Station to become the longest staying female on the space station.
Ooh, you go, girl.
Yeah, so we've got some exciting stuff right now. Some neat things going on in space. There's a lot of other stuff going on here on Earth, though. A lot of cool stuff. RoboGames is coming up in 2 weeks. Yeah. April 21st to 23rd in Pleasanton.
Those are always awesome.
My money's on the one with the buzzsaw.
Oh, you're taking the sucker money.
Yeah.
Yeah, that's. I'll take the other end of that one. It's the one with the low profile that doesn't have any big weapons that always wins.
Yeah, that's true.
It flips the other ones around.
Yeah. The flipper. You're right. It's always
the flipper. I don't know about that. We'll see. We will see how the RoboGames turn out. Who wins? Who will lose? You must go find your way at robogames.net if you're interested. Also, we are going to be in Philadelphia the June 10th and 11th. Right, we're going to Philly for the Young Innovators Fair. You can go to younginnovatorsfair.com to find information on that. All right, what do we have on the show tonight? I have stories about melting. Lots of melting. There's a lot of melting going on planet Earth. I also have giant viruses and cancer-fighting sperm. Justin. Yeah. Justin, what do you have?
I didn't realize I always wanted to be a doctor.
I'm on the edge of my seat, as it were.
Thought I had what it took. What did I bring? I've got early dino kin discovery, gut bacteria buffer against diabetes, disrupting electric-magnetic duality, and an answer to the age-old question, by the way, which one's pink?
Oh, I didn't know that was a question. Okay.
That's an age-old question.
Age-old question. All righty. And Blair, what is in the animal corner?
Oh, I brought nerdy bees, and I brought self-conscious elephants, and aliens living in our oceans.
Mm-hmm.
Aliens in our oceans. Man, all those sci-fis were right. I can't wait to find out more about this. We've got some good stuff coming up, so let's jump in. Get right down to it. So I'm going to start because, I was feeling a little spacey because of Yuri Gagarin at the beginning of the show. Did you all know that you can adopt the planet for Earth Day?
No, that's adorable.
Yeah, NASA is allowing us all #AdoptThePlanet. So we can adopt one of 64,000 individual pieces of Earth. And I tried to adopt the planet for This Week in Science, and let's see if I can pull it up right now. The location that I have is 3.6 degrees north. Oh, it's reloading, which is probably not. Yeah, 3.6 degrees north, 169.92 degrees east, which is somewhere way out there in the Pacific Ocean.
Yeah.
I'm not really sure much. But anyway, it gives lots of information about the location. So you can go to the web, you can look, it's through NASA. And if you look for this Adopt the Planet, you just put in a name of who you're adopting it for, and then it gives you data layers and information about that location. So in that location, it tells you relative chlorophyll levels, humidity, sea surface temperature, cloud top height information. And then it gives links and other things to be able to explore the map and find out more about different locations on the planet. And so really the idea is to make people more aware about what is out there on the planet and what different places are what's going on in different places on the planet, really. So it started on April 6th, and it'll be running through Earth Day. And you go to go.nasa.gov/adopt. And you can adopt a little bit of the planet, one of 64,000 locations. Ta-da!
Nice.
Yeah, pretty fun stuff. Also in NASA news, maybe not as exciting and fun, but researchers from the University of California, Irvine, NASA's Jet Propulsion Laboratory in Pasadena, and a bunch of other groups have made shipboard surveys of the seafloors in West Greenland. And they've also used data from NASA's Operation Icebridge and NASA's U.S. Geological Survey Landsat satellite. They've taken all the combined data and they have used it to generate the most comprehensive maps of the seafloor in the fjords beneath Greenland's glaciers that have ever, around 14 of the great glaciers. And this is the most comprehensive survey that has ever been done, like using all these different sources of data. And you know what they found out?
Everything's fine. Everything's cool here, people. No need to worry.
No, not really. So the previous estimates of the depth of the oceans in these fjords were as much as there were several thousand feet too shallow. So actually they found that areas of these in these fjords were actually much deeper than they had previously estimated. And, on first glance, you might go, oh, deeper ocean, colder water. That's awesome. But that's not really the way it works in this area of the ocean and the earth. And so because of upwelling and different currents, it actually, the deeper water sometimes tends to bring a lot more warm water closer to the glaciers. And so as a result of these changes in depths, and this finer grained resolution allows us better understanding of the possibility that these glaciers might be even more at risk of melting than they previously were. Well, the risk is not changed. Our understanding of the risk has changed.
Right.
Our expectation, our appraisal of risk. Yeah.
Yeah, exactly. So, so basically, the estimates of melting that we've had are probably not as rosy as they once were.
Right,
So you're saying we're in trouble?
I'm not saying we're in trouble. I'm just saying now we have more information.
So what you're saying is climate change is bad.
Yeah, we've spoken before, and this is one of the big things. We're talking about glaciers. We're not talking about icebergs. We're talking about ice that is on top of land. And if it melts, it's going to add to the oceans and will increase sea levels, right? That's bad. That's bad. So we don't want it to melt as quickly as warmer waters reaching the glaciers suggest that the possibility could make it.
If that happens, you're gonna have a bad time.
That'll be a bad day. Yeah,
I've yet to really hear one of these, information data adjustments to the model where it's like, oh, here's something that's really going to slow down the effects of global warming. It always seems like the more we look into it, the more we're like, our model was very rosy. The one that everybody was scared about, yeah, that one, very rosy. It's only worse news. All right.
Yeah, and so if we thought that Greenland, or at least our estimates of Greenland, were having a bad day, the Canadians and their freezers had a really bad day. Oh, so ice cores from the Canadian Arctic melted and left a big pool of water on the floor of a freezer in Canada. Yeah, so there was a cold storage facility in Edmonton run by the University of Alberta, and they had 180 meter long. That's about 3 feet for those of you who like to work in feet rather than scientific standards.
180 meters is only 3 feet? That's I've been doing my math all wrong. No,
shush, stop it. 180 ice cores, each of them about 1 meter in length. And they melted, and there was steam and water everywhere, and it was about 12 to 13% of the entire collection of samples that had been taken from across the Arctic. Yeah, and they lost. So researchers say some of the losses, one of the oldest ice cores from Mount Logan, which is a 5,500 meter high mountain in northern Canada. A researcher says we only lost 15 meters of the core, but because it was from the bottom, that's 16,000 years out of the 18,000 years that we had represented. Another one, they lost 66 meters of core from Baffin Island's Penny Ice Cap. That accounts for about a quarter of the record. 22,000 years gone, leaving a gap.
Guys, what's the one rule about ice cores?
They have to be frozen.
The one rule is that they have to be cold, you guys. Why was there not a backup generator?
I don't know. Yeah. So, that's an oops. That's an oops. This is the kind of thing. Each core would probably cost around $500,000 to $1 million to replace, according to the researchers, because of logistics with operating in the icy Canadian Arctic.
Yes, you have to make it warm enough for humans to not die in your research station, which means your ice cores may melt if you don't keep them cold.
Yeah, and this ice core archive actually has been moved around a whole bunch, and so it's moved from they were collected in the early 2000s, flown to Whitehorse, Yukon Territory, and then to the Ice Core Research Laboratory at the Geological Survey of Canada in Ottawa, and then moved to the Edmonton lab. So they've already moved around a whole bunch without ever melting. And then in a freezer,
Guys,
freezer
God,
goes.
some intern messed up.
I looked in the cooler and I saw it was all full of ice already, so I figured it was already cold.
Yeah,
It's
so-
cold with all that ice in there.
More like, Karen tripped over the cord getting coffee. Karen.
You melted the ice cores, Karen.
Karen, that's hundreds of years of research. How could you do this, Karen?
That's thousands of years of research. You're fired, Karen.
That's true.
Oh, yeah. So science, sometimes things aren't so rosy. They don't go as well as you plan them to because freezers go on the fritz.
I feel like that should have been in a freezer inside of a larger freezer that was inside of a yet larger freezer. And each freezer was attached to its own backup generator that also was attached to solar panels. That seems like the proper precautions for ice cores that are thousands of years old.
Yeah, so we've got ice cores, right? Difficult to get in the first place from glaciers and ice that are melting, that aren't necessarily going to be around forever. And they tell us a history of gases in the atmosphere of the planet. They give us a history of temperatures in the planet. They tell us all sorts of things about the history of our Earth. And yeah, if you melt them, no,
it
doesn't work so well.
It's frustrating too, considering the hard work it is to get ice cores.
And it's
the fact
very hard,
that humans have to trudge right on the back of essentially what is a snowmobile with a box tied to it.
Right.
And then they have to dig and then they have to core. It's a lot of work. And sometimes, researchers are out there for months. Ugh, what a bummer.
Yeah, but to be fair, Canadians probably spend as much time thinking about how to keep ice cold as we Californians think about how to make an iced tea warm. It's unintuitive thinking.
Should have brought in an expert.
Moving forward away from freezing and melting things, how about some more old stuff? I like old things, like giant viruses. We've talked before.
How giant?
Well, not, it's not like the giant that it's not going to be bigger than your house and coming to bear down on you like, virus. No, it's not like that. These are little tiny things, but big for viruses. Now, viruses we've talked about before is possible. People have hypothesized that maybe they're a separate branch in the tree of life, that there are these giant viruses, one that has been found called the Mimi viruses. They have these genes that potentially suggest that they can live outside of a host cell, that basically they could be their own almost a cell. They could exist on their own. They just don't have all of the stuff in them. And so some people think that viruses, these big giant viruses, they have a large genome associated with them. They're almost as big as they could almost be small single-celled organisms, except they don't have quite all the stuff that would be needed for that. Some people think that these viruses started out self-sufficiently and then got trapped inside other cells. And then they became, while they were trapped, they're like, I don't need all this stuff. I could use your stuff. And then they became, so then
there's all this biological material around. Why would I bother?
Get out of here.
Right. And so maybe, instead of becoming like a mitochondria, that they became their own little thing, kind of acting parasitically inside of small organisms. The other idea is that viruses might have been tiny bits of RNA, DNA that started grabbing bits of genetic material from hosts. So they never got trapped, but every time they encountered a new host, they'd be like, oh, I like that. I'm going to take that with me. Oh, I like that one. I'm going to take that one with me. And eventually, over time, that would allow some viruses taking this process of grabbing what they want and what they need to grow in size and become kind of big. And so there's a new study that's out in Science supporting the idea of not the parasitic entrapment hypothesis, but rather this, I don't know, kludge hypothesis. The kludge hypothesis, that it's just a bunch of stolen parts, a bunch of little bits and pieces.
What word are you saying?
Kludge, Kludge. Kludge. It's like the human brain. It's like things that evolution just kind of throw it together and it works. It's a solution that works. It might not be the best solution, but
I learned a new word today. Kludge, an ill-assorted collection of parts assembled to fulfill a particular purpose.
Oh, my life.
Just like your life.
Yeah.
My whole life. That's what this is. Yeah, good explanation. There's a word for that, huh?
My life is a kludge, and so are possibly these giant viruses in some total maybe. But it's, this paper has been based on a new group of giant viruses called Klosnu viruses, spelled Klosneu viruses. So Klosnu viruses is, I don't know if it's German or something. But anyway, it's debatable as to whether they are a missing link of sorts. And so these particular viruses code, their genomes code for lots and lots of enzymes and machinery that cells usually have to make proteins. And so that's super unusual. And they did a genetic analysis and they found that they've never seen anything like this. They have a high similarity to some single-celled organisms. And they found these new viruses. You want to know where they found them? In a sewage water treatment plant in Austria.
Yum.
So Klosnu viruses is probably Austrian. There we go, German. So they sequenced these genomes, and they're like, oh look, there's some genomes that are kind of similar to the giant Mimi virus that the French had found, and but the evidence, based on the kind of molecular clock and the genes that are involved, suggests that they could have originated in different ways. And so it's still unclear whether eukaryotic organisms whether the coronavirus took its bits and pieces from other organisms. They can't grow the virus yet. They've only been able to find it in samples. And so they don't have a host that they can infect to see what it does. And they don't infect, or they can't get it to infect the same type of amoeba as the Mimi virus. And so this is this new thing. I think, they found it in a sewage plant, so they should look for their host species in that sewage plant. There's something in that sewage. Otherwise, these big viruses wouldn't be there. But this is, it's just a very interesting finding.
Unless the host is the solution that they're in, I mean, if
the whole solution, right? Yeah.
In which case, is it out on its own at that point?
Right. So, yeah, it's very interesting. And so there's this virus. It has added a whole bunch of gene families that had never been seen before in the virus genomes. So it's got a whole bunch of new things, and they found the biggest thing here. They compared them to the Mimi virus, and they had been able to incorporate they can incorporate 14 of the 20 different amino acids into proteins without having to use host cell machinery. 14 out of 20, and that's a big deal. I mean, this is basically this coronavirus. It really seems like it's on its way to being its own its own cell, its own branch of life. But
so is that further back in the evolutionary spectrum than most of the viruses we see, or is it more advanced?
Oh, right. That's a really good question. I'm not sure.
Right? Because we don't know where viruses came from for sure yet. So we can't really know until then.
Yeah. Yeah. So it's anyway, very interesting that these viruses, maybe, I mean, still maybe they will tell us a thing or two about evolution and how life came to be, or why some bits of DNA and RNA decided to not become life and to latch on to life for the parts it needs to do things. And while other cells went, I'm going to be alive. It's a pretty neat comparison.
Mm-hmm.
Yeah. What you got, Justin?
I have something published in Nature. Scientists describe the earliest known dinosaur relative, 6-foot-long lizard-like carnivore called Teleocrater Verdenius. Says this is from Ken Englichik, the Field Museum's Associate Curator of Fossil Mammals, one of the paper's authors. Surprisingly, in quotey voice, earlier dinosaur relatives were pretty profoundly not dinosaur-like. Teleocrater has unexpectedly crocodile-like features that are causing us to completely reassess what we thought about the earliest stages of dinosaur evolution. So all dinosaurs are archosaurs, a group that contains dinos, birds, pterosaurs, that's the flying ones, and crocodilians. About 250 million years ago, archosaurs split into two groups, a bird-like group that evolved into dinosaurs, birds, and pterosaurs, and the crocodile-like group that includes crocodiles, alligators that are alive today, and a whole mess of now extinct relatives. This newly discovered Teleocrater Verdenius, I can't pronounce it, is the earliest member of the bird-like side of the family. However, it is not a direct ancestor of the dinosaurs. It's simply the oldest known cousin of the dinosaurs. It's sort of an offshoot branch right after the bird-like side of the family began. And it didn't look too much like a dinosaur. It more resembles a monitor lizard that would be alive today, for something you could picture in your head. It was between 6 and 10 feet long, although much of that length was neck and tail. It had really long neck and an even longer tail. Weighed between 20 and 65 pounds, was around 2 feet tall at the hip, and while some of its features, like the jaw muscle attachments at the back of the skull, clearly put it in the archosaur camp with the dinos and the birds, it had a lot in common with the more distant crocodilian cousins, notably ankle joints that could rotate from side to side as well as flexing up and down, whereas dinosaurs and birds' ankle joints can only hinge up and down motions. So it's got a more splayed crocodile gait than you might have expected from an early branch of the dinosaur family. So it's giving the scientists a new picture about how dinosaur-like features evolved. More quotey voice. We used to think that many of the distinctive features of birdline archosaurs evolved very quickly after they diverged from the crocodile line, because early birdline archosaurs were small and very dinosaur-like. Right? However, this one shows us archosaurs initially inherited many crocodile-like features from the common ancestors of all archosaurs, and that the typical birdline features evolved stepwise fashion over a long period of time, making Teleocrater a missing link, albeit from a slightly different chain, between dinosaurs and the common ancestors they share with crocodiles. And it's interesting looking at this, the artist rendition, at least of it, because I don't have a real one in front of me. You can see the both. Yeah, it's got crocodile-ish feet, and that sort of the neck and tail is this precursor to what we expect a dinosaur to look like. Very much so.
Mm-hmm. And the long jaw and lots of teeth for eating mammals.
Yeah. The name Tilley crater means slender, complete basin, a reference to the animal's lean build and closed hip socket, and is actually the name given to it in the 1930s when it was first discovered, although completely misplaced or misunderstood, or well, we were scratching the surface of dinosaur evolution at that time anyway. But they gave it the name that had originally been given to it. So that's pretty cool. But these are all new fossils. They found this is more quotey voice. We found fossils that we thought might be from Tilley crater, but it wasn't until we were back in the lab that we realized we found something really amazing. Discovery of such an important new species is a once-in-a-lifetime discovery. Tilley crater fundamentally changes our ideas about the earliest history of dinosaur relatives.
Right, so that potentially the Tilley crater came first, and then from there you have dinosaurs kind of coming beyond them. But before dinosaurs, you have the off-split of the actual crocodilian group.
Oh yeah, so we had
but this Tilley crater would be first because it has these crocodilian features.
Yeah, so he's closer to the ancestor of the crocodiles in some ways. But it's showing too that, some of these, you can't really call them crocodilian features then at that point. You have to say they're the precursor to the archosaur. This was something that didn't get discarded from the ancestor. Whereas some of the things that we think of as bird-like or dinosaur-like were things that were changed from the archetypal design that was there, right? Very interesting. And it's whenever we fit in one of these pieces of data that really changes how we have to assemble the rest of the puzzle in that time period, it's always very fascinating, because then we start to look at the whole process of evolution backwards with new detail, with new clarity, and then we can see that, especially in this case, those things are sometimes a loss of motion. That simpler, more primitive motion of the ankle, for instance, in dinosaurs and birds, may have come from an ancestor that had much more mobility, but didn't, over time, for whatever reason, the gait that they started using, the way that they started their locomotion, didn't require the mobility. We're relying maybe on teeth to do the grabbing and the biting, and so didn't need more mobile claws to grasp at things.
Yeah,
evolution doesn't always just add more benefits. Sometimes lets old ones go. Can't find use for it.
The thing that I'm always struck by, though, is that we're still looking at physical features. We're not looking at actual genetics because we can't get all the DNA. And so, we've even been surprised by genetic analysis of extant living organisms like birds. And genetic analysis reshuffled a lot of relationships between birds that had been determined on the basis of morphology. So, or how they look. And so all of this still, we need that genetic data, because we're still shuffling and reshuffling based on characteristics that might be convergent, that might have appeared for one reason or another. I don't know.
Yeah, morphology has proven to be sort of like a good outline.
Yes,
but it's not a final draft by any means.
Right.
It's somewhere at the very beginning of the process and then further from the end.
Kind of narrows things down at first.
Yeah.
Narrow it down. Oh, we're going through these stories one at a time. We're narrowing down our time. You know what time it is right now?
What time?
I think I do.
It's time for Blair's Animal Corner.
She loves our creatures, great and small. Biped, millipede, no pet at all. You wanna hear about animals? She's your girl. Except for giant pandas and squirrels. Animal Corner.
What you got, Blair?
I wanted to talk about animal smarts. One of my favorite things to talk about on the show. In general, we've learned What have we learned, Justin?
Birds are smart. So are a lot of other animals.
Yeah, in general, would you say that we have correctly assessed the smartness of animals?
We're getting there. The pack, really the one that blew me away, I think, more than any of the others was the pack hunting. Was it alligators?
Yeah, that's still one of my favorite stories of all time.
Yeah.
But so yes, animals time and time again surprise us at their intelligence, mainly because we still don't know how to test their intelligence. So I have a story actually sent by Ed Dyer, one of our faithful minions, about elephants that came out actually this morning from the University of Cambridge, and about elephant intelligence, elephant self-awareness, and particularly elephant body awareness. So a lot of animals that we try to study their sense of self is with a mirror test. There's a few problems with this that we've talked about a little bit before. One of them being not all animals understand what a mirror is. We just talked about that recently a couple weeks ago. Mm-hmm. Also, what if you're an animal that doesn't have a great sense of sight? What if you're an animal that doesn't have hugely different markings on each other? Like you all look pretty similar within a species, so it's just not something your brain's clued into, right? There's lots of things that can keep animals from being successful at a mirror test that is not a lack of self-awareness.
Right. Like part of it could also be animals are using a significant portion of the identification through the sense of smell.
Right.
And therefore, if the mirror doesn't smell like you wouldn't assume it was you.
Absolutely. So a recent study was looking at Asian elephants trying to find a new way of testing for self-awareness. So in this test, it was actually adapted from an awareness test for human children. The version for human children had them they had the kids push a shopping trolley, a shopping cart, right? But the cart was attached to a mat, and that mat had the child standing on it. So they couldn't push it if they were standing on the mat that the cart was attached to, right? So they had to figure out, oh, okay, so if I move off the cart, then I can move the cart, right? So in the elephant version of the test, they attached a stick to a rubber mat with a rope. The elephants they walked onto the mat, and then they were asked to hand the stick to the researcher. So they were previously taught to stand on the mat and hand a stick that was not attached to the mat, right? So they understood the task. In the results of the study, they found that they could figure it out. So actually, they stepped off of the mat an average of around 42 out of 48 times. So that's a pretty good success rate. And they only stepped off the mat in their control when the stick was not attached to the mat 3 times. So overall, looked like they weren't stepping off the mat by coincidence. They understood their bodies were in the way, so they stepped aside, which allowed them to do the task at hand. So these elephants are capable of recognizing themselves as separate objects from their environment, recognizing their impact on the environment, all that kind of stuff. So this is a new version of the self-recognition test that elephants pass with flying colors. We're not that surprised at that. Elephants are real smart. But now this gives us some tools for potentially testing other species in new ways about self-recognition. Additionally, this helps us look at body awareness of elephants in future studies in relation to social decision making. So if they know where their body is, we know how they know that their body is impacting a space. So that actually helps aid research on social dynamics with elephants and in space dynamics between elephants and humans. They actually are competing for land in Thailand and India. So conservation strategies require some research on this this conflict for space. So all of that could be helpful. But what's really fascinating here is that they really understood this test better than most toddler age humans. So they really could figure out how their mass, how their physical body was affecting the world around them in a way that was pretty intense.
I think it's very interesting, the idea of these elephants having this kind of body awareness. I mean, you kind of think it's like, of course they have body awareness. I mean, they've got a body. You have to know where your body is to be able to actually, you have to know where your body is in space to move and to do things and not injure yourself, not to fall in a hole. If you're an animal that lives in a tree, to not fall off a branch. You know, there are certain aspects of that awareness. Like if you're a bird, you have to know that you're landing on a branch and not, I mean, you can't look at your feet all the time when you're landing. You have to know where you're landing so that you don't miss it and fall, right?
Yeah.
But it's interesting that, you know, I was thinking about, you know, my own son and things that he did as he was getting older, as he became aware of his physical space.
Absolutely. And you wonder about some of it, like the bird. I wonder how much of that is based in riding a bike sort of thing. So if you learn how to fly, you learn how to land. If I asked you while you were riding a bike, where is your foot right now? Okay, where's your foot now? Okay, which way are you turning, right?
Yeah.
A lot of it is second nature. So where is this moment in which you recognize the difference between what you need to do what you're doing and awareness of where you are in space and time? And even we lose that from time to time, and we need to sit and think about where we are in space and time, right? So otherwise we would never bump into things, right, Justin? Like your sweet child.
Oh, yeah.
Your sweet daughter.
So if we were aware of where we were in space and time all the time, we might not run into doorways. So thinking about that.
Seven stitches. 3 years old, 7 stitches, 0 tears though.
Tell her to think like an elephant next time. Maybe
I know.
she will.
She continued
Be like
at
an
school.
elephant.
She was at school. She continued on to the she got up and went and sat down at the table to do a science activity of all things.
Yeah.
And like everybody was just staring because she was, you know, playing around there and had blood coming down.
Head wounds are always
She just
bad
wanted
for
to
that.
science. So yes, so these elephants.
Who cares about the bleeding? I just want to
my gosh.
science.
So that's a
Far
case
enough away
where
from my heart. I'll be fine.
where being smart, maybe for your daughter as well, is a huge benefit, right? But being smart isn't always a good thing, is it?
Yes, it is. It's always a good thing. Why would it not be a good?
Let me just quickly remind you, for any of our listeners out there, I'm not saying that any of us know what it may have been like, but being a smarter person in class, sometimes maybe you get picked on. Maybe life's a little bit tougher for you.
What school did you go to?
Well, if you're a B, there is a definite drawback to being smart. Bees
being smart,
being too smart. So bumblebees, research from U of G. I don't know what that means. University of
Georgia,
and also the Plant and Food Research Conglomerate Organization. There's no noun at the end there, in New Zealand. These two groups, they came together to look at how smart bumblebees are. And ultimately, they found that fast-learning bumblebees collected fewer resources in the colony over their foraging career. And there is a learning-associated cost in the wild, therefore, which means actually it costs to be smart. So the way that they tested this is they had blue and yellow artificial flowers in a lab. They had 85 individual bumblebees from 5 colonies. The yellow flowers provided sugar reward, the blue ones did not. And then the researchers observed how long it took the bees to ignore the unrewarding blue flowers. So initially, they would prefer the blue flowers because that would be their natural inclination. So how long it took them to adjust and instead associate yellow flowers with food. The bees were monitored using radio frequency identification, tagging technology to determine their foraging activity. And they found that the bumblebees that were best at adjusting actually came at a cost. So they found that they got less resources and overall were less successful as bumblebees, even though they were better at learning. So this is weird. It's hard to parse out exactly what's happening here, but as far as they can tell, it all comes down to the cost of being smart, the actual energetic cost of having a smarter brain.
Yeah. I don't know if I buy that explanation. It's an interesting correlation. I just think those bees are self-aware and they're just, man, this is it. I'm part of this hive mind. I'm going to have to do the same job every day. Never going to move up. Can't be the queen.
It's interesting you say that, Justin, because actually that's part of the story. Part of the story is that these bees are still here. So the idea is that neural tissue is metabolically expensive, and it's also metabolically expensive to maintain throughout the life of the bee. Foraging is energy demanding, so is learning. So the kind of the give and take here, the push-pull, is that if you take energy to make and produce and maintain a good brain, you will have less energy to forage. The question then is, why, oh why, are there still smart bees?
But I wonder if, you know, if you've got the waggle dance, right? You've got a bee that comes back and tells the other bees where the nectar is. Where do you have to go? And bees that pick up on that more quickly are going to be able to get to that location more accurately. They're not going to wander off track. They're probably going to be more successful over time.
So that's exactly it too. But also,
that wouldn't be my. I would actually expect. Hang on. I would actually expect the bee that does the waggle dance to be the smarter bee. That's the one who's like, look, I can give you really good, clear directions. I found it really quickly. I didn't spend time foraging. Figured I'd just come back to a real accurate map of where it's at, and all of you drones can go do the actual work. Like I'd love to see that. Like. If they're the ones who tend to report back more, the smarter bees.
So, okay, so based on this study, they found that the bees that learned which flowers to go to not only foraged less successfully, but had a shorter foraging lifespan. So they double lost. The idea behind...
What do you mean by less successfully? I mean, I thought that it was just that they didn't go on as many foraging bouts, that they didn't forage as much.
They didn't forage as much, and they didn't forage for as long of a lifespan.
Because they didn't need to.
Because they didn't need to.
They did better.
They had existential angst. They had too much self-awareness. This is going to be my life forever.
So
every day, the same thing. Monotony. I don't take it anymore.
So as far as I know, the bees were bringing back the same amount of stuff regardless. So the fact that they're going out less, that most likely means they're bringing back less total. But the reason they think this maintains in a bumblebee population is the same reason that that you still want that nerd that everybody beats up on in class. You still want him around because everyone needs to copy off his paper during the test. So the smart bees are actually helping to rise up the average intelligence level of the bee population as a whole. So the population has an advantage based on these outliers helping bring everybody up. So they help lead the way, they help assist in the foraging strategy overall. So even though they lose out, they are helping the population.
I see that happening. Yeah.
So there's downsides to being smart, but maybe you can save your colony.
So there's a population level upside. that keeps the smart bees around.
Well, I want to study the dumbest bee now and see, do they live really long life or are they dead even sooner?
Well, we talked about lazy ants, so I bet it's probably pretty similar.
These social organisms. And did you have one more story for the animal corner?
I'll save it for later.
Wait, what? This is an exciting story. Okay.
Okay. All right. I'll do it now. I'll do it now if you want me to.
You can do it in the second half.
You can do it in the second half. That's cool.
It's gonna be a bit, and I know it's time for a break. So
it's going to be a bit.
It's a dense story.
All right. Well, we will take a very short break for everyone to clear your heads and think about smart bees, dumb bees, all the bees around. And
you're a dumb bee. I'm not a dumb bee.
Dumb bees, smart elephants. Let's think about these things as we move into the break. Everyone, this is This Week in Science. We'll be back in just a few moments. We have some messages for you. So stay tuned for more This Week in Science. More science stories coming up in just a few. Hey, everybody. Don't forget, April 21st through 23rd, RoboGames is happening. Go to robogames.net if you are interested in finding out more about the RoboGames and the festivities happening at the Alameda Fairgrounds in Pleasanton, California. Also, mark your calendars if you are near Philadelphia. This Week in Science is going to be broadcasting live from the Young Innovators Fair. You can go to younginnovatorsfair.com for information on that event. Also, if you are interested in helping produce TWIS, if you want to help TWIS happen every week, you can help us out by donating or by buying our merchandise. We have a whole bunch of stuff. If you head over to TWIS.org, our website, our wonderful portal to the world of TWIS, just head on over there and go to the main header bar. And if you're interested, the Zazzle store. You click on that Zazzle store link and it will take you to our Zazzle show, our Zazzle This Week in Science page, which has all of the products that we have made available, hats and mouse pads and lumbar pillows, even mugs, sweatshirts, phone covers, polo shirts, necklaces, stamps. We have a lot of stuff with the logo for TWIS emblazoned all over it. So you can let people know about TWIS just by wearing something or putting a stamp on an envelope. Kind of neat that way, right? And additionally, you can have wonderful Blair's Animal Corner original artwork pieces too. Those lumbar pillows are pretty cool. There's wrapping paper, there are ties, all sorts of neat things. So head on over to that Zazzle store if you want to support TWIS, because part of the proceeds, part of the sales from all of these items comes right back to TWIS to keep the show running. So you can help us produce TWIS just by buying some of our stuff. If you're not into stuff and you're a minimalist, I get that totally. Why don't you try clicking on our Patreon link? Patreon is also one of the links in our main head bar over at twis.org. Click on that link. It'll take you to our Patreon community page where we are collecting patrons. You can become a patron of the TWIS Arts. That's right. We produce our show every week, and you can subscribe or become a patron on a weekly basis, on a monthly basis. You set your limits, set your amounts, and it's a neat little community where sometimes we send out videos to you before they're released to everybody else. You get special content, special letters that other people don't get. It's kind of a neat place to be able to support TWIS and help us produce our show. But if you don't want to if you're done with social networks, just click on our donate button. Go to twis.org, click on the donate button. It won't make you join a social network. It'll just make it really easy for you to donate money to TWIS. And if all this is not your cup of tea, we totally get that too. You can help us out just by telling people about TWIS. If you can tell people to go to our website, twis.org, where they can find the latest podcasts and they can listen to them, or if you tell them to go to iTunes, where they can subscribe to our podcast, or the Google Play Store, they can also use that podcast directory to subscribe to our podcast. All sorts of places that we can be found. Spreaker, SoundCloud. We are out there, and we would love you to let people know about us. Tell people, if you're a YouTuber, if you're watching us on YouTube right now, share the YouTube link. Let people know. If you watch us on Facebook, share it on Facebook. Send people our way, because the more we grow, the better it's going to get. The more we'll be able to do, and the more science fun we will all be able to have together. You are important in making this show happen. We really couldn't do this without you. Thank you for your support.
Can't believe what a skeptic I am. I can't believe you believe in that sham. We disagree, but I still give a damn. The ramification of treatments from holy men leaves me slightly queasy deep down in the abdomen, convinced that the lives that they lead need adjusting.
And we're back with more This Week in Science.
All right, we are back. Justin, what else did you bring?
I brought two groups of participants that participated in a Finnish diabetes study. All of these participants were overweight and had impaired glucose tolerance. So the researchers investigated the serum metabolite profile. Serum it's your blood if it didn't have any blood cells or coagulating things. So it's the broth. I don't know how to describe it.
It's the thing that supervillains always use.
Well, yeah. So they looked at the metabolite profile of these 200 participants with impaired glucose tolerance who either developed type 2 diabetes within the first 5 years or did not convert to type 2 diabetes within a 15-year follow-up. So these are all high-risk type 2 diabetes category folks, pre-diabetes. They tracked them. They studied their the serum, their blood, to see what would happen. So they're not being given a serum. It's not like these are control groups in that way. They're just looking to see what happens in these groups. They found that a high concentration of indole propionic acid in the serum protected against type 2 diabetes. Indole propionic acid is a metabolite produced by, wait for it, intestinal bacteria.
Oh.
And that intestinal bacteria has its own production boosted by a fiber-rich diet. Higher concentration of the acid also seems to promote insulin secretion by pancreatic beta cells, which might explain the protective effect that it's having. According to the researchers, the discovery provides additional insight into the role of intestinal bacteria and the interplay between diet metabolism and overall health. Findings were published in scientific reports. Differences between the groups were analyzed by non-targeted metabolomics analysis. So
metabolomics.
Basically what it means is instead of having gone in with, we're going to look for this, this, and this, and see how that tracks over the time, what they did was they just looked at the overall picture. And what they saw were spikes in this acid for those who didn't develop and lower levels in those who did develop. So they just looked at the overall stuff that was in the serum and didn't prejudge it. They weren't tracking anything specifically going in. So they didn't go in with a premise or hypothesis. This is what we'll find. They just observed and took their observations and made connections between their observations and the results, right? They also found that certain lipid metabolites were concentrations of that, as well as the indole propionic acid had the correlation, right? So they also took this out of this one study And they looked at 2 other population-based data sets. One was the Finnish metabolic syndrome in men study, and another was the Swedish Västerbotten intervention project. So these are 2 other different data sets. And they found this acid was also in more abundance in the protected groups in those studies. So a nice sort of open sourceness between these studies allowed them to take their results that they got out of their study, look into other studies that were taking place, and went, aha, well, the same thing is present here. So they also identified several new lipid metabolites whose high concentration were associated with improved insulin resistance and reduced risk of diabetes, less low-level inflammation, and the like. Concentrations of these metabolites were also associated with dietary fat. Lower amount of saturated fat in the diet, the higher the concentrations of these metabolites. Quotey voice, earlier studies too have linked intestinal bacteria with the risk of disease in overweight people. Our findings suggest that indole propionic acid may be one factor that mediates the protective effect of diet and intestinal bacteria. It's from Academy Research Fellow Kati Havaneva from the University of Eastern Finland, which is almost unpronounceable.
We'll have to get Yanni from Finland to pronounce it for us.
Hanhineva. I'm pretty sure it's Katie Hanhineva from the University of Eastern Finland. Direct identification of intestinal bacteria, they say, is a complex process, which is why identifying the metabolites produced by intestinal bacteria may be a more feasible method for analyzing the role of intestinal bacteria in the pathogenesis of diseases, for example, diabetes, than it would be into just figuring out what their beneficial factors are, right?
Yeah, I think we've talked so much in the past about how fiber in the diet is beneficial for bacteria. And now we're finding that, not just looking at the bacteria, but this particular substance, the indole propionic acid, that this is involved and that it's a signaling molecule somehow with the pancreas, and that all these factors potentially work together to be protective for some individuals. And, in the case of people who maybe have diets low in fiber, that protective effect is not there because the bacteria are not protected and given a good environment by fiber in the diet.
Right. So within this one study, we pretty much go from eat your fruits and vegetables, right?
Right.
To high fiber is good for intestinal bacteria that produce this acid that incites the pancreas. So like the chain, the mechanisms start to unfold now.
But the result is still the same.
Yes.
Eat healthy, everyone.
Yeah. Weight loss, exercise, dietary adjustments, whole grain products, fruits and vegetables.
Oh, and like sleep and
Right. Yeah, but I mean, we all know, we love French fries. French fries are yummy. Potato chips, sugary sodas. I mean, I can't drink them. They're too sugary, but there are all these things that, we love them. I love ordering French fries instead of a salad, but moderation, right? Make sure you have that salad on occasion. Make sure you're working through that and not just always ordering French fries.
And it's funny you say that because kind of like my takeaway from this story a little bit is like how to hack it. Right? It's like this is things like, okay, now we understand the mechanisms as they cascade down and become catalysts for other things. How do we still drink soda and eat French fries and not get diabetes? Like this is sort of like once we hack the system, we don't have to just stick to the exercise diet thing anymore. Like we can cut around it somehow.
Right.
At least.
And potentially this is, this indole propionic acid is maybe something that can take the place, of salads eventually.
Right.
But that's not good either, because this is still correlation. This is we see these people either converting to diabetes or not converting to diabetes, right? Same kind of profiles, but it's this acid that kind of made the difference, right? So it's still correlational. They haven't actually gotten this causation, like complete causation. There could be other
there's
factors.
Also these lipids involved, which they also did see. And the other thing too is, the more we learn about anything,
the more there is to learn.
Yeah, the more we learn about any of it, we start to find that it's a community of catalysts. It's an ecosystem of things taking place, and that you may be able to, like, you suggest, just add this acid into the mix and not get a result, even though it's essential to it. If the rest of those factors, like when we were talking about milk last week, right? In the cancer, right? Like it's still probably a very important part of this process of cancers developing, but might, or obviously isn't, a sole operator. So yeah, all of these things are very complicated, and that's what's fantastic. The more we drill down on them, the more we can figure out, suss out what individual actors are doing within these grand plays.
So don't fly off the handle and eat a salad yet. Wait.
No. That's what you can do. You at least know where this starts, with salad, exercise, fruits, vegetables.
That's where it starts. Yes. Eat your fiber today, folks.
Yeah. But in the future, they're going to live off
fiber a day keeps the diabetes away. All right, everybody. Moving on forward, let's talk about sperm and how researchers are creating sperm that will have the power to fight cancer.
I cannot wait. Tell me everything.
This is I love this study. So Mariana Medina Sanchez from the Institute for Integrative Nanosciences in Germany have created this, basically are using sperm as a drug delivery system. And so they're loading up sperm and
just because they're the best swimmers in the world.
Because they got that flagellum. Demonstrate away, Blair. That's
back in the room.
Yes.
In the room. I know I haven't raised my hand all semester.
Yep. What's your name?
Where
what
exactly are they delivering to?
Yes. So what are they delivering to? This is designed for diseases, cancers of the genital tract, the urogenital tract. So the female reproductive tract is the site of many cancers: uterine, cervical cancers, endometriosis, inflammatory diseases of the pelvis, lots of things that can be very problematic, and it's very difficult to deliver drugs specifically to that area. And so the system...
tell me about the delivery system.
They have put the sperm into a harness. They put a little harness on the body of the sperm, and that harness is coated in iron. Okay.
So it's like a sperm with a backpack on, is what you're saying?
Sperm with an iron backpack.
Okay. Great.
And then they have basically got these sperm they soak the sperm in the drug. They have an active ingredient. They just soak it in the active ingredient and then the drug. And so the sperm they soak up all the drug, and they've got it attached to themselves all over, like a little, got little, lots of drug all over themselves. And then the harness with the iron harness.
Okay. So they're wearing a drug jumpsuit and an iron backpack.
There you go. Okay. Yes. And then they use magnets in an external magnetic field to steer the sperm to the site of delivery.
Okay. Again, though,
because iron backpack, magnetic, and then the sperm are like, wee, I'm swimming, and magnets.
What is the delivery system?
The sperm, and then
how is the sperm getting there?
Okay, so the sperm is directed there by the magnets. The magnetic field kind of directs it. The sperm has the propulsion system to drive it forward. And so if you know where a tumor is, say in the cervical wall or in the uterus, you can potentially have this, with the magnets, have the sperm swim right up there and specifically target the tumor, swim right up to it. And then the harness has a quick release on it. And so when the sperm bumps up against something, the tumor, whatever it is, the harness breaks away. It's like a breakaway harness. And then the sperm can swim away on its own. And so then the sperm can reach the tumor, harness is released, and then the sperm will burrow into that tumorous tissue because sperms like to keep swimming and burrow into things. And so then they would enter the tissue where the cancerous cells are located and deliver the drug.
Okay, so I guess this is what I'm not getting. So the sperm, is it from a donor? Is it from?
Yeah, so I don't know where you would get sperm.
I know.
It's not.
I know where it comes from.
It's not.
I can answer
it's not
that
artificial sperm. So this sperm is still it's normal sperm out to do the normal job of sperm. So,
so there's a problem.
Pregnant when you're treating someone with this special sperm.
Yeah, you're right. You're catching the right point there, Blair. That is the one problem that the hurdle they haven't overcome yet with this drug delivery system. You might get rid of your cancer, but you might also get pregnant.
Great.
So the other thing is, I'm surprised that the sperm would. I mean, maybe I shouldn't be, but that sperm will just penetrate anything they run into.
Yeah.
That just
pretty
burrow, and they break down after a while. They have a lifespan of a certain amount of time, and then they start deteriorating and breaking down. But
rest after a long swim like that.
Yeah, they get tired. They only have a certain amount of energy. Little get up and go.
Everybody.
Yeah, and
they're taking sperm. They're outfitting them with the drug jumpsuit and the iron backpack in a lab, in a dish, in a dish.
Tube. This hasn't been tested in people yet.
So it's not like they're giving men supplements that then create them to produce sperm with drug jumpsuits and iron backpacks.
Yes, no, there's none of that. No, this is not that
yet.
Not yet. Yes, but I mean. The what's going on there,
Have
Justin?
your husband take these.
Hello, ladies.
Go have a good night and call me in the morning. Right?
And then
Dr. Justin is here to cure what ails you. No.
Nope. Yep.
I don't think that was even implied in anything that Kiki said, Blair. I think you just came up with that.
Yeah. I did. You're welcome.
Yeah, so there's still a few questions about this, but you can read about it yourself on the archive server, preprint server, archive.org, titled...
I almost feel like at the end of it, you just did one of these.
The study is titled Sperm Hybrid Micromotor for Drug Delivery in the Female Reproductive Tract.
AKA sperm with jumpsuits and backpacks.
Sperm fighting cancer.
I think I always picture sperm with backpacks, like parachutes. I think it's from a Woody Allen movie.
Yes.
Yeah.
That it is.
I think that's still my...
All right. Tell me about electrodynamics, Justin. Is that your next story?
Yeah, this is crazy. Radio waves, microwaves, light itself. They're all made of electric and magnetic fields. Classical theory of electromagnetism. They completed that theory back in the 60s by, James Clerk Maxwell at the time.
Ooh, and you can even use induced magnetic currents from an electrical, magnetic fields from an electrical current to tell the sperm what to do.
Yeah.
So this goes right along. There you go.
So at the time, Maxwell's theory was not thought to be used on sperm, but was still revolutionary. It provided a unified framework to understand electricity, magnetism, even optics, Now, new research led by LSU Department of Physics and Astronomy Assistant Professor Ivan Agulo with colleagues from Universidad de Valencia, Spain, is advancing our knowledge of this theory. Recent discoveries have been published in Physical Review Letters. Maxwell's theory displays a remarkable feature here. It remains unaltered under the interchange of electric and magnetic fields when charges and currents are not present. This symmetry is called electromagnetic duality, right? Amazing. Only thing is, when electric charges exist, magnetic charges have never, while electric charges exist, magnetic charges have never been observed in nature. So while this is a beautiful theory and everything seems to work, one of the fundamental principles has never been seen. So how does that, if magnetic charges don't exist, the symmetry doesn't exist. So one of the underlying fundamental ideas behind this theory doesn't exist.
Wait, one of the ideas behind a theory doesn't exist.
Yeah, one of the fundamental ideas behind the theory that seems to work just fine for everything that we've used it for, which is everything that you're hearing me, seeing this show, every technology, every bit of electricity that's running around in people's homes these days. It's all based on something that may not exist.
So this all works, but now we don't know why.
Yeah.
Well, we haven't ever known why.
We haven't ever known why.
We thought we knew. Turns out we don't know anything.
We've never been able to prove it. And physicists have been motivated to search for these charges, magnetic charges, magnetic monopoles. However, nobody has been successful. And Agulo and his colleagues may have discovered why we have been successful. Quotey voice. Gravity spoils the symmetry. Regardless of whether magnetic monopoles exist or not, this is shocking. The bottom line is that symmetry cannot exist in our universe at the fundamental level because gravity is everywhere.
Darn gravity, that thing that we know but don't get.
Gravity, right now, this is another thing. Gravity exists, but how does gravity work? That we also don't know. But it says here, gravity, together with quantum effects, disrupts the electric magnetic duality or symmetry of the electromagnetic field. Agulo and colleagues discovered this by looking at previous theories that illustrate this phenomenon, among other types of particles in the universe called fermions, and applied it to photons in electromagnetic fields. We have been able to write the theory of the electromagnetic field in a way that very much resembles the theory of fermions, and prove this absence of symmetry by using powerful techniques that were developed by fermions. This new discovery challenges assumptions that could impact other research, including, that's drum rolly sound, the birth of the universe.
We're taking it all the way back.
That's the Big Bang. Satellites collect data from radiation emitted from the Big Bang, the cosmic microwave background. This radiation contains valuable information about the history of the universe that we base the length of the age of the universe on, and the expanding universe, the lights, it's all these things. By measuring CMB, we get precise information on how the Big Bang happened. Scientists analyzing this data have assumed that the polarization of photons in the cosmic microwave background is not affected by the gravitational field in the universe, which is true only if electromagnetic symmetry exists. However, since this new finding suggests that the symmetry does not exist at the fundamental level, the polarization of the C and B, or cosmic microwave background, can change throughout cosmic evolution. Dun, dun, dun. Scientists may need to take this into consideration when analyzing the data. And focus of Agulo's new current research is on how much this new effect actually is. So, right now, there's not being called into question, but there's an asterisk ding, right there on the age of the universe and the timing of the Big Bang if all the data in needs to be adjusted.
So what we thought was a constant is not a constant. So
Right.
just...
And well, I mean, we do pretty well. We do pretty well just looking at distant galaxies, and that's sort of the number.
Yeah, I'm sure we're not far off
for the age of the known universe. But pinpointing how much further back in time the Big Bang took place, if there is adjustments over, many billions of years, the 5%, 10%, 20%, like these numbers can make a very large shift over that kind of time frame. So exciting, really fantastically exciting. This means we're going to have more to talk about in the future. I love the unsettled question that we thought was settled, because it's like, oh, we no longer need to talk about the age of the universe. It's just a number. That's just textbook reading. We won't cover that here on This Week in Science anymore. And then, that's going to be future stories.
Yeah, but this is one of those things that I think is, even though this new idea has come out, and they've got maths underlying this determination, there have been lots of maths underlying other determinations about gravity and our universe, and different hypotheses on how things work the way that they do in physics, right? How does certain phenomena work? And so I think what this is going to do more so is it's going to really fire up the debate about symmetry, and especially in this electromagnetic instance. And if nothing else, we will see more conversations come out of it.
Yeah, and I would also, it's not debate also for the it's not debate in terms of, well, I believe this, but you believe that, so let's argue about it. It's going to be more maths. It's going to be reviewing maths,
that's what
testing it
about.
What are my assumptions? What are your assumptions? Because assumptions make an ASS out of you and me.
Right. But so science is all about getting new information and adjusting what we know, right? And the things that are not science will not accept new information and refuse to adjust, right? That's what makes science different. That's what makes science great, is the adjustment and the conversation.
And I think it's also important, like in this study or in this research, they weren't setting out to disprove the age of the universe. They were chasing the maths on a particular subject, and they came to a conclusion that by its existence challenges other data sets.
Yep.
And those data sets may need a good shaking up. We all need to shake up our data sets from time to time.
Shake up your data set. What that means is take a look at it again, reassess, make sure that you are looking at things with a clear perspective. Yeah. And as we try to look at things with a clear perspective, it gets harder to see things clearly as you get older, right? Brain gets tired. You've seen too many things, had too much experience. Oh, why don't we just go ahead and die already? No, wait, I don't want to die. And we've had
Kiki, this show just got really dark all of a sudden.
I don't want to die. So maybe
I hope this is a segue. Otherwise,
it is a segue. That's what I'm working on right now. So, modern technology, scientists are working on, looking for factors in the blood of young mice to determine, whether or not they're going to keep old mice fresh and
old mice. Does your mice get old and stale? You want to make your old mice fresh and new again.
Yeah, give it a young mouse blood transfusion, right?
That's right. There we go. So we've talked about that before on the show, and this study is another one right along that vein. But it takes our other favorite, which is fecal transplants. Puts it right in there, except it's not fecal transplants. It's fish eating the poo of younger fish.
Ooh.
Yes. Yes. So this is another that was posted to the bioRxiv preprint server, so it still needs yet to be reviewed. But this study out of the Max Planck Institute for Biology of Aging in Cologne, Germany, has looked at fish and looked at a particular species, Nothobranchius forzeri, the turquoise killifish. And they determined these fish live in ephemeral ponds. Those are ponds that pop up during the rainy season in Mozambique and Zimbabwe. And these fish will eat, they eat whatever's around. And so they did a transplant of gut microbes from 6-week-old killifish into middle-aged 9-week-old fish. And they had also cleared out the natural flora of the middle-aged fish with antibiotics. And then checked to see what happened. And so these transplanted microbes recolonized the guts of the fish, and dun dun dun, the microbiomes of the middle-aged fish turned into the microbiomes of the 6-week-old fish. So in effect, they had this really young gut, and the fish lived longer.
Huh.
Yeah, so their longevity increased by 41% compared to fish that had not been exposed to the microbes.
Okay, so I know what's next.
6%.
Rabbits, because rabbits eat poop.
Right?
Introducing Dr. Justin's Not a Real Doctor new line of baby poo pills.
Baby poop pills.
Yeah. 41% longer life as seen in different species.
Yeah, and so they found that the older fish, when they did this experiment in reverse, the microbiota from the older fish did not affect the lifespans of the younger fish at all. So unlike what we've seen in the blood experiments, where blood from old mice actually affects the metabolic profile of younger mice, this is not what happened. So in the older poo, the older gut microbes, didn't affect longevity at all. So
and if I could speculate a little bit,
yes,
I bet you, those younger fish have a virus load that was doing proper filtering, and that probably
like the story from last week.
Yeah,
that's what I would speculate. No, that was from a long time ago, but that's what I would speculate.
Yeah, I don't know if it's we don't know if it's a viral load, but the researchers think that it has to do with the immune system. And their guess is that as you age, your immune system gets kind of tired. This is why as you get older, allergies tend to be less severe than when you're young. You don't have as severe reactions to why colds and flus can be more dangerous. So what they think is that harmful microbes, when you're older, can outcompete the good microbes. And by getting a microbiome transplant, it could have reset the microbiome of the middle-aged fish. And so what you would expect is that if it's an immune system thing, that over time, the middle-aged fish microbiome would deteriorate to a more, or a population that represents less beneficial bacteria. Yeah. So anyway, they're going to look into things a little bit more. And they want to know more about, here we are, fish, people, mice, poo transplants. Mm-hmm. There we go. Oh, to be young. It just takes the blood and the guts. The poo.
To be young and also a fish.
And also be a fish. I know. Yeah. All right. We should talk to our friend who did the microbiome transplant.
Josiah.
We should talk to Mr. Zahner and see how old his friend was.
Oh,
comparatively.
yeah.
I think they were in college together, though.
I think it's probably the same age.
Yeah. So that wouldn't have an effect. All right, Blair, you have a big story.
Yes. Do you want to talk about aliens that live in the ocean?
Mm-hmm.
I'm talking about octopuses. Sorry.
They're not aliens?
They're not aliens, but they're like aliens because they do so many things in such weird ways. It seems like they were just dropped off here. It's so unusual. And the list of ways that they are so unusual is starting to stack up to a ridiculous degree. Their weird camouflage, the fact they have 9 brains, all of these, their brain is shaped like a donut. They can fit through anything larger than their beak. They're just bizarre animals. But on top of this, now recent research, octopus brain. So research looking at the RNA editing of cephalopods is bonkers. So a few years ago, actually, I reported on the show about cephalopods that were going through RNA editing while in cold water. So this is essentially some sort of Lamarckian-esque evolution happening where they were changing elements of themselves, changing the coding of proteins in real time within individuals. From that discovery back in 2012, people have started looking in closer and closer at these oddballs of the natural world and their DNA and RNA processes. So this new study, which is coming out of Woods Hole in Massachusetts, suggests that the evolutionary path to neural sophistication, so the way they got so smart, includes a huge amount of RNA editing. When I say huge amount, I mean in our bodies, a fraction of 1% of our RNA have recoding events, have events where they change, where they switch out an A, a C, a G, or a T. They don't follow the DNA so that they edit. They essentially change the way the protein is formed. That happens less than 1% of the time that RNAs are coding. In these cephalopods, over 60% of RNA transcriptions.
60%?
60% of RNA transcriptions are recoded by editing. What does this mean? What this means?
It means they're going to outlast every other living life form on this planet and eventually take over.
It means they can adapt to anything.
We already know they're on the whole doing pretty darn well in the face of climate change and ocean acidification. We already know they found a lot of ways to survive in extreme environments. And now they can edit themselves in real time to face new challenges. So cephalopods, they got under control. So they looked specifically in this study at 3 smart, quote unquote, cephalopod species, 2 octopuses and 1 cuttlefish, after they had done the editing in a squid brain. So now they were looking at squids, cuttlefish, and octopus. These are called coleoids. So they're the soft-bodied cephalopods. So that does not include nautilus, basically. Poor nautilus, left out. I know. But so these guys are considered to be extremely smart. And now we're thinking that that's how. But what's especially interesting is that this has happened at the expense of what we consider to be normal evolution. So when you have an animal rewriting DNA, more than half of the time, that's the norm. That means that inherited traits are often dwarfed by these edits.
But so it's not DNA they're editing. They're editing RNA.
Right. But the RNA is what then makes proteins. So even though they're inheriting
expression.
Right. So they're inheriting certain things in their genes, but those genes are not actually dictating what happens in their body because there's this editing that happens in between. And so they're saying that this is actually affecting, this is impacting the effectiveness of evolution in cephalopods. And so instead of being an organism that mutates via evolution or changes via evolution and then occasionally has editing events, it's completely the other way around.
So it's really, really slow genomic evolution, and everything else is changing.
Yes.
But this is where the epigenetic memory thing can kind of come in, because are the options for the changes based on environment, or however the RNA editing is taking place, is that inherited? Is that something? Is that like a toolbox? That's
good question. So because it's happening at the RNA level,
pull that one out. If you see that, this one goes in its place.
So because it's happening at the RNA level, it shouldn't be inherited. Because it's happening at the RNA level, it's not happening in the DNA. The DNA is what's passed on to future generations. So this is only happening. So basically, the little zipper RNA is coming along the double helix and copying what it sees and saying, okay, there's an A, okay, there's a G, okay, there's an A, okay, there's a T, and then, okay, here's a U, okay, here's a T, and then, oh, I see an A, but you know what? Screw that. I'm going to put an I in here instead. Okay, moving on, right?
But what triggers, this is the thing though, what triggers, and it's usually an environmental factor of some sort that triggers the, as you said, screw it maneuver. and having that big of that flexible of a toolbox is obviously inherited. Otherwise they would have just seen it in one, right? So this is an evolution, like
overall, to answer your question, this is overall in response to temperature changes. So it is when the environment is not what they are normally adapted to. This is how they adjust to new temperatures. And so this is what the study back in 2012 found, was that these I think it was an octopus that was living in either it was Arctic or Antarctic waters, but it was at a pole. It was ridiculously cold. This octopus should not have been able to live there at all, but it was doing it via RNA editing. And that's what kind of spurned this whole study. But it's interesting because this is something that, as far as we know, unprecedented in biology. As the research paper said, this goes directly against the, quote, central dogma of evolution. So it's bizarre. Will we see it in other groups of animals? I don't know. My money's kind of on no, because cephalopods are real weird. Like I started with, they are so unusual. We just found out recently that they might actually be colorblind, even though they use color camouflage and signaling. And so they're using the halo effect around their oddly shaped pupils to detect changes in color. Another bizarre thing. Have we seen that anywhere else in the animal kingdom? Absolutely not.
Actually, we have seen that. We have seen that in the human kingdom. When they first invented a way to see TV in color, there were these screens you would put in front of the black and white television that would assign colors to things, although it wasn't always.
Well, us humans are good at engineering things that are not as they are, but yes, that is the first time we've seen it in the natural world in animals. So cephalopods, man, they're going about it a different way, and they'll be here when we're all gone.
I feel like I've long ago, at one of the early episodes of TWIS, predicted that the cephalopods would eventually inherit the Earth.
Mm-hmm. It's possible. I mean, we're gonna, overfish, climate change. It's gonna be jellyfish, cockroaches, and cephalopods. Yeah. That's what it'll be.
Yeah, and it's so funny. I was at a soiree for NOAA, for the National Oceanographic Atmospheric Association in the Bay Area. They had an octopus soiree, which is how I actually heard about this press release originally. And there was an octopus researcher who spoke, and she said, octopuses are going to outlive us all. Yeah. So there you go.
That's super exciting. We'll be outlived by the octopuses. Not if we can get our hands on those fecal transplants and blood of things.
And I get my singularity. Where's my robot?
Where's my singularity? Right. Where can I download
And I
my brain?
learned this just after the youngest child is out of diapers.
Final stories for the evening. There are crazy parasites in Hawaii. It's parasitic roundworm. Causes rat lungworm disease. It's been in Hawaii before, but it usually doesn't cause that many cases. But in the last few months, there have been 6 cases that popped up. This doesn't make it an epidemic necessarily, but it's a parasite of concern. And the question here, is this what's going on? Why is this particular parasite starting to expand and become dangerous? The problem with it, and it's also spreading to places like Georgia, or no, not Georgia, Oklahoma, various states around the country. There have been instances of this rat lungworm caused by this roundworm. The roundworm, Angiostrongylus cantonensis, can be contracted basically by eating slug larvae or residue left by slugs. And this is usually because you've had raw fruits or vegetables or had unclean drinking water. So you want to make sure
slugs.
Yeah, so you want to make sure
is like the main subsistence of being on Hawaii.
Exactly. So you want to make sure things are washed really well. Make sure things are very, very clean and washed. And make sure you're drinking clean water. So this infection has been introduced to North America through globalization. So it's one of the things is people have thought it's gotten to the Americas through ship ballasts, which we know is a big issue for the spread of many things.
How is this still a problem? Ballasts.
I know we've had this conversation. Yes. So globalization is a big cause of its spread, and then also climate change is making environments better for it to survive and be passed along. And then finally, this story was sent to me by Michael McThenny, actually sent to TWIS on Twitter by Michael McThenny. Crabs in trees.
Huh?
What?
Tree crabs.
Tree crabs, yes. So this is Kenny Maranjandu, and it was found in the forests of the Western Ghats in South India. And it's named after the Kani, a tribal community who first noticed the crabs. And Maranjandu is the local term for tree crab. So basically the Kani tree crab. And so these long-legged crabs were first reported in 2014, but then a researcher from the University of Kerala was like, I need to go look for these. Let's go actually find them as opposed to hearing these anecdotal stories. So they tracked the tree crabs down and have just published in the Journal of Crustacean Biology about these crabs. These wonderful tree crabs that have long legs, a distinct hard outer shell, and they can get they live in water-filled hollows of tall evergreen and deciduous trees. And the Kani, who look for them and will probably eat them, detect their presence by looking for air bubbles coming out of the hollows of the trees. Yeah. But they're cute, dark colored little crabs. And I mean, seriously, watch out for coconuts and crabs. Falling coconuts, falling crabs. It's pretty cool. Tree crabs.
Oh gosh, they look spidery.
They do look spidery, right? Well,
crabs and spiders are pretty.
They're related, right? Yeah, exactly. They're very related. They're cute little crabs. Cute little tree crabs. They're not that big as your hand, maybe. Cute. You don't like it? You don't want the Blair, I thought you would love the tree crab.
I love the tree crab. I'm being a tree crab.
Okay, be the tree crab. Love the tree crab. Yeah, crabs and trees. Tree crabs. There we go. The habitat, it's very restricted in its habitat. So anytime you have an organism that's restricted in its habitat, you have to think about the ecosystem and threats to that ecosystem. The existence of these crabs is reliant on rainwater that falls and collects in the tree hollows. And so if rain goes away, then the crabs themselves will have a hard time existing in the trees. What was your last story, Justin?
My last story is about a pistol shrimp or snapping shrimp. There's a new one discovered. These types of shrimp have the ability to generate a substantial amount of sonic energy.
Oh, I love those.
By closing the enlarged claw, it has one enlarged claw, at a rapid speed, the shrimp creates a high-pressure cavitation bubble, the implosion of which results in one of the loudest sounds in the ocean, strong enough to stun or even kill a small fish. This particular one, found off the Pacific coast of Panama, has an extremely pink enlarged claw.
Ooh.
And the scientist that discovered this particular shrimp also has a favorite band. Quotey voice, I've been listening to Pink Floyd since The Wall was released in 1979. I was 14 years old. I've seen them play live several times since, including the Hyde Park reunion gig for Live 8 in 2005. The description of this new species of pistol shrimp was the perfect opportunity to finally give a nod to my favorite band, says Arthur Anker, University of Federal de Goiás, Brazil. I think is at the and also Kirsten Hultgen, Seattle University, and Sammy DeGrave, Oxford University Museum of Natural History. They all it's DeGrave. Sorry, it's DeGrave. Sammy DeGrave, Oxford University Museum of Natural History, who's a lifelong Pink Floyd fan. So they gave the name Sinalphius Pink Floyd.
Sinalphius Pink Floydia.
Sinalphius Pink Floydy.
Pink Floydy. Sinalphius Pink Floydy. I love it. What was it? A week or two ago, we were talking about David Attenborough getting yet another species. Yes. Finally one for Pink Floyd.
Finally, about time. How's no one named a shrimp after Pink Floyd yet? I mean, come on.
It's the first crustacean, but they have other stuff named after him. There's a damselfly that's named Umaguma.
Umaguma.
Yeah, that's one of the albums.
That's fantastic. Oh, when pop culture comes in, intersects with science.
I mean, what's the point of finding new species if you can't name them silly things?
Right?
Yeah, I feel like a lot of these cases, they're like, well, I named one after my advisor, and I named one after my wife, and I named one after my son, and I named one after my university. Right. Pink Floyd? Yeah, Pink Floyd. Yeah,
Pink Floyd. This guy found the guy who discovered like the tiniest snake ever discovered, named it after his wife, which it sounds sweet and endearing until you find out he had named 28 other critters before that. It's like, what took you so long?
Yeah.
Like, what were the 28?
Who were the 20?
That next
got
snake,
around to your wife.
That next snake you find, you'll be naming that after the couch you'll be sleeping on. Damn.
Well, it is time for us to wrap up this show, and I know Justin has a couch to sleep on, so I would like to remind everyone once again, enjoy this evening. It's Yuri's night. It's auspicious in the history of space flight. Comes around once a year, and tonight is that night. Thank you, Yuri Gagarin. Also, RoboGames, April 21st to 23rd, and the Young Innovators Fair in Philadelphia, June 10th and 11th. Check these things out if you are interested in finding out more. We'll have information at our website. I would love to give shout outs, say a word of thanks to our Patreon sponsors at this point in time. Thank you to Chris Clark, Paul Disney, G. Burton Lattimore, John Ratnaswamy, Richard Onimus, Byron Lee, EO, Jared Lysette, Kevin Parachin, Andy Groh, Keith Corsell, Jake Jones, John Gridley, Stephen Bickle, Kevin Railsback, Gerald Sorrells, Ulysses Adkins, Derek Nichols, Dave Friedel, James Randall, Eric Schwab, Bob Calder, Mark Mazzaros, Ed Dyer, Trainer 84, Brian Hedrick, Layla, Marshall Clark, Charlene Henry, Larry Garcia, Randy Mazuka, Tony Steele, Gerald Onyago, Steve DeBell, Greg Guthman, Brian Staub, Patrick Cohn, XV, Daryl Lambert, Haroon Sarang, Melissa Mosley, Alex Wilson, Jason Schneiderman, Dave Neighbor, Jason Dozier, Matthew Litwin, Eric Knapp, Jason Roberts. Richard Porter, Rodney, David Wiley, Robert Aston, Todd Northcutt, Arlene Moss, Aurora Lee, Bill Kersey, Ben Rothig, Darwin Hannon, Rudy Garcia, Felix Alvarez, Cosmic Gypsy, Brian Hone, Orly Radio, Brian Conran, Mark, Nathan Greco, Hexator, Mitch Neves, Flying Out, John Crocker, Christopher Dreyer, Artyom Shuwada, Dave Wilkinson, Steve Mashinsky, Rick Ramos, Gary Swinsberg, Phil Nadeau, Braxton Howard, Sal Good, Sam Metzetter, Emma Grenier, Philip Shane, James Dobson, Kurt Larson, Stefan Insum, Honey Moss, Mountain Sloth, Jim DePoe, John Maloney, Jason Olds, James Paul West, Alec Doty, Alumalama, Joe Wheeler, Dougal Campbell, Craig Porter, Adam Mishkan, Aaron Luthen, Marjorie Paul, David Simmerly, Tyler Harrison, Columbo Ahmed. Thank you for all of your support on Patreon. And if you're interested in supporting us on Patreon and being a producer of TWIS, you can go to patreon.com/thisweekinscience for more information. And remember that you can always help us out simply by telling your friends, family, coworkers, people on the bus about TWIS. And on next week's show, we will be back once again with more science, broadcasting live online at 8 p.m. Pacific time on Wednesday. You can catch us at twis.org/live to watch and join our chat room. But if you can't make that, don't you worry. This show is archived for posterity at twis.org/YouTube and also just at twis.org and on Facebook. It's all over.
We're there too. Wow. Showing up everywhere. Thank you for enjoying the show. TWIS is also available as a podcast. Just Google This Week in Science in your iTunes directory. Or if you have a mobile type device, you can look for TWIS, the number 4, Droid app in the Android marketplace, or simply This Week in Science in anything Apple Marketplace.
For more information on anything you've heard here today, if you want to know more about that fish poop or that sperm with a jumpsuit and a backpack, show notes will be available on our website. That's at www.twis.org. That's TWIS.org, where you can also make comments and start conversations with the hosts and other listeners. Maybe you want to post a picture of a sperm in a jumpsuit and a backpack.
Or maybe you just want to contact us directly. Email Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or Blair Baz at twis.org. Be sure to put TWIS, TWIS, somewhere in the subject line, or your email will be spam filtered into oblivion. You can also hit us up on the Twitter, where we are at TWIS Science, at Dr.Kiki, at JacksonFly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know.
We will be back here next week with some more amazing imagery, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, hey, remember,
it's all in your head. Why?
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer and it shouldn't be news. That what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. Jeopardy. And This Week in Science is coming your way. So So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. Cause it's This Week in Science. This Week in Science. This Week in Science. Science. This Week in Science. This Week in Science. This Week in Science. Science. I've got a laundry list of items I want to address. From stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. So how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say. And if you learned anything from the words that we said, then please just remember it's all in your head. Cause it's This Week in Science. This Week in Science. This Week in Science. Science. This Week in Science. This Week in Science. This Week in Science. Science. This Week in 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 Science.
And now I unmute myself from the dancing muting, and it is our after show. Time for the after show. Stick around if you'd like to. This is not the show anymore. This is where we're just hanging out. I don't know how long we're going to hang out, but this is just the part where we hang out and we talk to each other sometimes, every once in a while. Except Blair is tired because she had a big dinner last night.
It was the second night of Passover, so I had a Passover seder.
Big Passover seder.
20 people.
That's a lot. 20
Which
people is a
means
lot.
Cooking a lot of food, but also cooking a lot of very particular food.
Mm-hmm. Yes. I had Taco Tuesday.
That's nice.
Yeah, we had some friends over for Taco Tuesday. That was nice. I like Taco Tuesday. Taco Tuesday.
We have to book flights for Philadelphia, don't we?
Yeah, I'm not going to book them yet, though. I mean, it's still a little ways off.
But that's exciting.
Yeah, I mean, I've been contacted by them once in the last couple of months because they're like, oh, we're going to start our marketing stuff. Can you send us some stuff? And I was like, okay. But then I haven't heard from them since then, really. So, I'm going to hedge on the side of, we're going to tell people about it, but I'm not going to buy any tickets until I'm pretty sure.
Good call.
Yeah. We're gonna, I'll buy tickets at an optimal distance, a month ahead of time or something. So May, that's soon. How is that so soon?
That's what I was saying.
Why is this
soon.
year going so fast?
It's going so fast. I literally just started writing 2017 on things, and now it's April. It's halfway through April. It's not even just April. It's mid-April.
I know. I really don't like this phenomena wherein time goes faster the older you get. I do not like it at all.
I do feel like last year moved a lot slower.
Yeah. Yeah. I don't like it.
It's crazy. I think it's relation to how busy you are, right?
Mm-hmm.
I guess how many things you have to do.
I guess that means I have a lot of stuff to do.
I'll tell you what.
I'll tell you what.
Yeah, how's your year going, Justin?
How's your year?
I'll tell you what.
What.
One thing that slows down time is encountering things that are new. when you encounter something new, it's not routine. It's not the in and out of the day. It's not the busy process of doing XYZ task. And that's what this show does. This show slows down time because every week I'm encountering.
That's true, actually. Yeah.
You know, when I'm reading over the stories, when I'm getting prepped for the show,
it's being present, right?
Yeah, it's being
notice.
Really, but it's also with really new material every week, and I hope on some level that listening to this show has the same effect. Throwing something into the diet that's not just a formula of media consumption. This is the setup to the joke. This is it's going to have a conclusion. Oh, there's the conclusion to the joke, or whatever it is. You know, the day that you spend doing maybe slightly different each day, but largely the same processes at work. You know, it's time to do this. The alarm goes off. It's time to wake up. You have this many minutes to get ready. All of that for me slows down completely when getting ready for this show and reading stuff I never would have encountered throughout my normal week and haven't seen anywhere else. And I really sincerely hope that some of that effect translates to the audience, that they slow down time a little bit while listening to this 1 hour.
This 1 hour of programming
that sometimes drifts into 2. That by encountering new information, new stories, finding out more about this world we live in, that helps slow down other people's journey as well.
The TWIS meditation. Yeah. So how's your daughter? She was tough. She was bloodied.
My daughter's fine. Yeah.
Not yours, Blair.
No tears. Like she did a great job of, being calm the whole time that we were doing the stitches and stuff. She was playing right up to it. And then when she finally didn't have to sit still anymore, was a bundle of energy that was running around playing again.
That's great. What a trooper.
Didn't phase her at all.
It's almost as if women are really good at handling pain.
There was one moment, like after, like
the child, the resilience of child.
She had to go potty. She hadn't told anybody she had to go potty until we were done. She's like, okay, now I got to go potty. So it's the first thing we did after stitches, and she caught her the reflection in the mirror, and she had, as they were kept cleaning the wound stuff, there was sort of blood splatter all over, and she's like, I look like a vampire. Like I'm drinking blood. I'm like, yeah, totally. Like the sight of her own blood, she's totally unfazed by anything. And I already knew she was a pretty brave kid, but this was definitely the biggest test of that I've seen, and flying colors. I'm very proud.
Awesome. Have you seen the Thor Ragnarok trailer?
Oh yeah,
so many people are talking to me about that. I haven't seen it yet.
So good. I can't wait to see it.
I got tagged by that.
I did that.
Yeah.
I couldn't help it.
Why? Is
Justin
in it?
The only one.
Justin's in it. Yeah.
Yeah. Jeff, my alter ego.
There he is. I gotta find the image so that I can.
My alter ego.
Alter ego. Exactly.
Jeff Goldblum's in it.
Possibly my actual biological father is in it, and therefore everybody was suggesting that telling me that I've gone gray, basically, I guess, because he's sports some gray hair.
Jeff Goldblum is in Thor. Are you serious?
Darn it.
Oh yeah, he's like the centerpiece of it.
Hold on, I'm gonna find this. I'm gonna find a picture so that you can. Are you watching it?
Yes.
Are you watching it? Yeah. Oh, there it is. Come on. There it is. Paused. Okay. There we go. Here we go. So, we will. I'm not showing the whole trailer, but here we go. You need to start wearing gray hair and lower lid eyeliner.
And lower lid eyeliner. That one I've got already.
You've got a little lower lid eyeliner, and then there's the blue chin stripe. But the hair, we got gray hair now, so you have to work on that, Justin. And start dyeing your hair.
Oh, second. No.
Oh, there he is.
Look, there will come a day.
Dada.
There will come a day that I figure, I don't know, I might decide to diet. I have yet to do this.
Diet, like change your diet.
No,
Yeah, how is Loki still alive? This doesn't make any sense.
I know. I need to
he's
go
immortal.
back and
What do you mean,
watch.
how is an immortal still alive? The immortals are immortal.
I love it. Every time there's like a new movie in a series, it makes me go back and watch the other one again because you have to go watch the other ones so you know what's going on in the new one. Things happen, like memory loss.
He's a friend from work.
I know. Oh, I know him. He's a friend from work.
Yeah, Genescu, I do need a throne. I should replace this shoddy chair that I've got.
Oh, wait,
Yes,
that's
a
great. I
throne.
can't wait
There
for
we
that.
go.
It looks amazing.
Great. He's gonna need like a cloak, a cool coat.
Yeah, so
Yeah.
you know what your Halloween costume is now, Justin.
Mm-hmm.
It's pirate. It's always pirate.
You have to brush in the gray
It's always just pirate.
and then dress up your children as
Jeff
people from
Goldblum, the
Thor.
pirate.
No,
Yeah.
like one of your children can be Loki. You'll make the stupid crown out of like
That'd be great.
gold tinfoil.
Someone can be Thor.
Yeah. Great.
Yeah. Yeah, Whiskey Renegade Marvel's doing pretty well for themselves in the movie business.
Mhm.
They certainly are.
Okay. I'm gonna exit stage left.
Yeah. Is there anything else we need to talk about before you exit? Are we
I don't know.
good? Everything's good.
I think so.
Everything
Hmm.
under control.
No news?
Everything is under control, except for
just-
the fact that nothing is under control.
Does our producer have any notes for
We're
us?
gonna. That's after. We're gonna have to do an after the after show real quick for that one. If there's.
I don't have any notes.
So
no.
for this.
Great.
No notes.
Oh, in that case, say goodnight, Blair.
Good night, Blair. Say good night, Justin.
Good night, Justin.
Good
Say
night,
goodnight,
Kiki.
Good night, Kiki.
Wait, what did I say? What did you
You
say?
said say good night. You're supposed to say good night, Kiki.
No, it's supposed to be say. Last time we said
No.
Good night.
and then I said.
Oh my God.
It's supposed to be.
Kiki, on the show that you weren't here.
No,
we
no.
talked
I'm right.
forever.
You're wrong. We just messed
Planning
up last
this.
week.
No, it was say good night, Blair. Good night, Blair. Say good night, Justin. Good night, Justin. And then 1, 2, 3. Good night, Kiki.
Okay.
And then you would say
you're
good
right.
night, and then it'd be over. See? That's what we were
Start
supposed
over.
to do.
Start over. Try it again. We'll fix it in post.
Okay, you do it.
Say goodnight, Blair.
Good night, Blair. Say good night, Justin.
Good night, Justin. Good night,
Good night,
Kiki.
Kiki.
Aw, good night, everybody. Thanks, everyone, for watching. Sorry it's such a short after show. We've got some tired Blairness going on. We got some tired people. But we will be back next week. We hope you have a wonderful intervening week. For those of you who are hunting for Easter eggs this weekend, I hope you find them. That's great. There you go. We'll see you next week. Tell your friends about TWIS. Bye. Or just hit the button, Kiki.